I will introduce today's speaker. Veronica Brown is the lab manager at UT Genomics Core, and she's going to be talking about the support that they offer to the UT research community. So with that, I will turn it over to you, Veronica.
Thank you. I'm really excited to speak to you all. I saw a couple names already logging in of people I recognize, at least one of whom I've already done some sequencing for, and someone who I think I have a meeting with coming up soon. So that's super exciting. I will put the caveat out there that I just got done teaching a 3-hour class. I am in an extra classroom. And so if we get interrupted, I've told people I'm using this room for this purpose, but I don't guarantee we won't get interrupted. But I'm going to do my best. And if this is not my best presentation at coming up with words, forgive me since I have been teaching before this. Seems like everybody wants all their meetings at noon on Thursdays. All right. So for those of you who are not familiar with core facilities at the University of Tennessee, core facilities are shared resources available to UT researchers at a cost recovery basis. So what this means is we're not out trying to take more money from you than we need to. We are just trying to recover our costs. Depending on what the lab is, sometimes that means usually it means we're recovering researcher salary, but usually can include consumables, instrument fees, things like that. These facilities offer access to high-end instrumentation, technical support, expert consultation to the UT research community, and in most cases, external customers as well. All core facilities are becoming centralized under the Office of Research. And if you are interested in more core facilities, there is the link to the OREAD core facilities page. I like to start with a slide on where I'm located. I'm in the Science and Engineering Research Facility, SURF, building on top of the hill. My lab is easy to find. It is room 407, and we also have some smaller labs on the 4th floor, rooms 402 and 404. My office is hard to find. If you see the map, the picture down in the bottom, it is a door that looks like a closet on the main floor back behind the stairs. I've described it to someone once and they said, oh, like Harry Potter's closet. And I love that explanation. So it's my little Harry Potter office. It really was not meant to be an office. It looks like a closet. But if you walk in the main doors, it is straight across. This picture needs updating. They're painting it. It is not quite that bright anymore. But unfortunately, the 4th floor still got yellow, so it's still pretty bright. I do keep my office door locked all the time because I am in the foyer, so you will have to knock to get my attention. Other core facilities that are located in SURF, there are 3 of us. And the 3 of us actually work really well together. So my genomics core, we offer high throughput DNA and RNA library prep and sequencing on Illumina instruments for UT researchers as well as off-campus researchers. There is also the mass spec lab that offers gas and liquid chromatology coupled with high resolution mass spectrophotometry for analysis of biological small molecules. And you have the contact information for both those cores if you need it. And then there's also Advanced Microscopy and Imaging Center, where they have services and instruments for both light and electron microscopy. In the microscopy lab, they do a lot more training to let users use the microscopes on their own. All right, some more about what we offer in my lab. We do DNA and RNA extraction. We do library prep of amplicons, genomes, metagenomes, and RNA-seq. I'm going to go into all of these in detail. And we also offer custom projects with consultation. So whether or not we take on custom projects depends on a lot of things. Some projects make sense for us to take on and some don't. Usually that comes down to cost. Some of the custom projects we take on, users may start them and we come in part way and take over. Most library preps end up doing very similar preps toward the end. So it makes sense for us to take over when it becomes a common use prep. Sometimes it's as easy as if you buy the kit, we'll take over and do it, or we'll do it with you. And then we offer high throughput sequencing on Illumina iSeq, NextSeq, and NovaSeq. These are the instruments you see there on the right. Now, I love this picture of all of them together, but it is a little misleading because they are opposite in size there. The iSeq, we're going to go into detail on these in a minute, but the iSeq at the top is the baby sequencer. It's actually quite small. Then there is the NextSeq in the middle, which people describe as looking as like an air fryer. And then at the bottom is the NovaSeq, which is my beast, and it looks like a refrigerator. We can come in at any point in the process. We can do extractions, we can do library preps, or we can do what we consider load-only samples that are ready to put directly on the instrument. We consider all samples to be BSL-1. We can take in BSL-2 samples, but we need to know ahead of time. My lab is considered to be BSL 1.5, which is a term environmental health and safety made up for us because we can take in BSL-2 samples, but we don't need any of the paperwork associated with them. That's up to users. And if we have any of the samples left over after we do the extraction, we're going to ask you to come back and get them. For my purposes, samples are only BSL-2 for the very first step. Once we've extracted the DNA and start working on the library prep, they're usually, I'm going to put safe in quotes, but they're usually fairly safe and benign for us to work with. So when thinking about project design, our lab currently only offers high throughput next-gen sequencing. That doesn't mean Sanger sequencing is not of use anymore. It depends on what your project needs are. Sanger sequencing is more traditional sequencing, what students and more traditional projects may be used to. There's still benefits to Sanger sequencing. It has a familiar workflow. It's generally fairly easy, and it can be cost effective when you're only sequencing a small number of targets. The challenges are it has low sensitivity, it's low throughput, it does not have as high of discovery power, and it's not cost effective for a large number of samples. It is also getting more and more expensive to do, period. That is why we don't currently offer it here. Next-gen sequencing has a high sequencing depth, which enables really high sensitivity. It has high discovery power, high mutation rate resolution. It's massively parallel sequencing, enables you to do a lot of samples at once, which brings the cost down a lot. It allows you to detect a lot of variation. The challenge is it's not effective for simple detection of a low number of samples. That's where I come in. I enjoy helping people figure out what is the right type of project for them. Is high throughput sequencing right for them? Sometimes the answer is no. I don't want to say I turn people away, but if high throughput sequencing is not right for you, I'm going to help figure out what is right for you. Common next-gen sequencing workflows. Obviously, the first thing we have to do is isolate the product. That should be the easy part. That's not always the easy part. I will tell you that myself and my technicians all are good at isolating DNA and RNA, and we have a background in difficult samples. I myself am a product of UT. I've been here for more years than I will put in a recording. But my background is in the ecology department. I started in a bat research lab, and the majority of my work there was on bat fecal samples. And I learned how to do those extractions before there were fecal DNA kits, so I did them manually. I consider that I am good at the hard stuff because I started before it was easy. So we're really good at the difficult extractions, and if you're having trouble with them, we'd love to help you. That's where we shine. The majority of the time we come in at the library prep stage, and we'll talk a lot more about that as we go on. Then obviously the sequencing is where we come in. We have the high throughput sequences in the area. We will also talk a bit more about where Oak Ridge comes in, but the biggest instrument, the NovaSeq, Oak Ridge has some of the smaller instruments, but we have the big workhorse of the NovaSeq. The reason we have that and not Oak Ridge is it's a lot easier for Oak Ridge users to get to us than for our users to get to Oak Ridge. It does not make sense for both of us to have that big instrument because neither one of us would be running it at max capacity, and it's a very expensive instrument. We do not offer data analysis, but I have some slides at the end of some labs we collaborate with to help with data analysis. All of the instruments that I'm going to talk about work on a patterned flow cell technology. I could spend an entire hour talking about this because I find it super exciting and really interesting. All of the sequencing takes place on a little piece of glass, a glass flow cell. The new technology looks like a honeycomb. There are only certain spots on this glass flow cell where the DNA can bind. I say DNA. We sequence both DNA and RNA, but by the time you sequence RNA, it's been converted to cDNA. So when I say we sequence DNA, if it is RNA, it is cDNA. So we always sequence DNA. On these honeycombed glass plates, you see the little grooves there. That's where the DNA is going to fall in and bind. This means that it's a lot easier for the instrument to find the spots where the DNA is, and it makes it harder to make mistakes. This is a very complex slide. If you are interested in how the technology works, I encourage you. It sounds horrible, and as a teacher, I hate this answer, but Google the YouTube videos of how sequencing by synthesis works, and it explains this slide in motion. The nanowells are where the DNA binds. Then you see in that red slide pictures of the DNA actually binding. There's lots of spots within those wells. Hopefully, only one fragment of DNA has bound within the well. Then it moves down to the pink slide where it's made many copies of this DNA, and then it's ready for sequencing. There is a lot going on in this slide, but it is actually my favorite slide because I think it really simplifies how this sequencing works. The title of this slide, Library Prep is Critical for Successful Sequencing. That says a lot. Oftentimes I have labs that don't have a lot of money that can't afford the kits. Many times the mistakes I see in sequencing happen when people try to do this on their own. Every single piece you see down at the bottom must be in your final product, and it must be there to the base, or it's not going to happen correctly. So at the top half of this slide, you see a very simplified graphic of how these protocols work. You're going to start with some sample DNA. Somehow you're going to break up this DNA into smaller fragments. All of my sequencing technologies are short read fragments. We need small fragments. You're going to either ligate or through PCR, put adapters on these small fragments of DNA so that they will attach to the flow cells. Almost always there's going to be some kind of PCR amplification, usually some kind of short cycle PCR. There are non-PCR options. They're not my favorite. They don't work as well. Something users don't like to hear, but if you don't have much DNA going in and you use a non-PCR option, you don't have much DNA to put on the flow cell. That sounds obvious. People don't like to hear that when they have not as good a quality samples. While the PCR-free protocols have their benefits, they also are a lot harder to work with. But either way, you're going to bind the adapters to your fragmented DNA and hopefully amplify it a little bit so that you have a lot of copies. Then down at the bottom, you see a layout of what the pieces are. Now, on the red and light green fragments that say P5 and P7, those names P5 and P7 don't matter that much. Think of those as flow cell primers. Those are the pieces that are going to match to the flow cell to attach it. Those are on the ends. Those are going to attach us to the flow cell. Then the index 1 and index 2. Those are what are going to allow us to multiplex and pool a whole lot of samples at once, which are where we save money. Then the one that say RD1 and 2 SP, those are read 1 and read 2 sequencing primers. Those pieces serve a whole lot of purposes depending on what library prep you're doing. But once it's on the flow cell, their job is to make the pieces sequence. It's where the sequencing starts on the flow cell. Again, I could spend the whole talk just on this slide, but we're going to move on past this one. I'm happy to talk more about this later if anyone is interested. So the sequencers that we have in our lab, I get super excited about these. I absolutely love the instruments we have in our lab. One of the reasons I started reaching out to different groups to try to give talks like these is because we have two new instruments. The first one is the iSeq. I will tell you these names get really confusing, not the iSeq so much as the next two. The iSeq is my baby sequencer. It can give you 4 million read pairs or clusters. Now that terminology is also confusing. Illumina uses the term clusters. What we mean by this is in those honeycombs, that's one spot where a DNA fragment is going to be sequenced. I like the term read pairs. So if you think about that, if we are sequencing in the forward and reverse direction, that number is going to be doubled. So it's a read pair. We can get 4 million read pairs, spots where we're going to sequence DNA. Now, the downside of this instrument is that it can only sequence 150 bases in each direction. The most common thing we use this instrument for is testing bigger projects before we're going to put it on a more expensive run. It costs less than $1,000 to all users. We have a tiered structure that we'll talk about, tiered pricing structure that we'll talk about later, but no matter which tier you're in, it's less than $1,000 to sequence on this instrument. So if maybe you have a really big project that costs thousands and thousands of dollars, I'm probably going to encourage you to run it on this instrument first, just to make sure we got it working. Now, the one in the middle, oh, I should say, down the right-hand side is the different flow cells. If you come take a tour of my lab, I'll show you these flow cells and how beautiful they are. The NextSeq is one of my new instruments. This is my favorite instrument by far. It is the easiest to use. It has the best current technology. It has two different flow cell options. They're color-coded. One is purple, one is orange. One can give you 100 million read pairs, the other 400 million. It has a read length of up to 300 bases in each direction. When I say it has good quality, I absolutely mean it. I am not just saying that. I literally do happy dances when I see the quality coming off of this instrument. If any of you were familiar with the MySeq said it could sequence up to 300 bases. It lied. The quality coming off of that instrument was not good out to that long. The quality coming off of this instrument, as long as you put in good quality samples, the quality off of this instrument is beautiful all the way through 300 bases in each direction. Now, since we have this iSeq, the baby sequencer, and the NextSeq, we're kind of missing those in-between projects. However, we have quadrupled that amount of data, but lowered the price. The price for sequencing an equivalent run that you might, well, not equivalent because you get 4 times the amount of data. The smallest run you might put on the NextSeq compared to what you would have put on the MySeq, 4 times the amount of data for less money. And if you only needed the size of a MySeq, I might be able to pool your project with someone else and save you even more money. So even though we're missing that 25 million read pair project that went on the MySeq, I'm saving you money. So if you have a project that you think might work on the NextSeq, come talk to me. I get really excited when I talk about this instrument because I truly love it and I love the quality coming off of it. Then there is my NovaSeq. I still love the NovaSeq. The NextSeq is my favorite. I try not to say that out loud in front of them. The NovaSeq has a whole lot of flow cell options. Those are the bigger ones in that picture, the big white ones. When people need a lot of sequences, that's when we start talking about the NovaSeq. It can read up to 10 billion with a B read pairs. Now, most people don't need that many on their own, but some of the people doing human work, plant work, they do need a lot of reads. The NovaSeq, because it has a lot of flow cell options, it gets really complicated really fast. It can read 2 by 250 bases, but only on the smallest flow cell that produces the least amount of data. And when I say least, it is 800 million. So it's still a lot of data. That one's really good for amplicons. All of the other flow cells that get up to 10 billion read 2 by 150. So those are good for RNA-seq and really big genomes. The benefit of this instrument, it is really easy and really useful for pooling people's projects when you have a common read length, such as 2 by 150. When people come to me with the big projects, I'm going to ask you, how much data do you need? And do you want that data fast, or do you fast and maybe a little bit more expensive, or can you wait a little longer till I can pool it and save you more money? The greatest cost savings is when we can put it on these bigger flow cells, but maybe it might take a little bit longer to get it pooled. The way we get things moving faster with this pooling is I need more people to bring samples that they're willing to wait just a little bit longer. And the more people we get sending samples in for pooling, the faster we can get everybody's turned around. That last bullet point on there, every February, Illumina redoes their prices. This price period, they gave me a huge discount on NovaSeq reagents, and my prices for NovaSeq sequencing just dropped dramatically. And that is not just for internal users, it is for ORNL users as well. So please, please, please come talk to me because I'm really excited about our new pricing. This is just a summary slide of all the different instruments. So remember, the iSeq is the baby sequencer, mostly for testing. NextSeq, I don't hide the fact that it is my favorite, even though it's not the biggest. Excellent quality. The NovaSeq gives good quality too. I don't belittle the quality coming off the NovaSeq, but I love that we can get up to 300 bases off of the NextSeq. And then the NovaSeq is the best for getting the cost down lower when we can pool projects. All right, that's the technical part. Hopefully for those of you that get bored by that, I can pull you back in now. I love that part, obviously. So common library preps that we do. Whole genomes and metagenomes. So a whole genome is when you have one individual, so maybe one plant, and you're going to sequence the genome of just that plant. A metagenome would be where you have a mixed community, such as soil, and you want to sequence, I'm going to put in quotes, everything from that sample. For me, that library prep is the exact same thing. Our lab does a whole lot of amplicons or meta barcoding. That's a whole lot of PCR products. I did not realize until recently that our lab does a lot more of that than other cores. And I think that's from my background that I am really good at getting tricky PCRs to work. And then since we now have the sequencers that can do a larger depth of sequencing, we are getting good at RNA-seq as well. So I'm going to go into a bit of detail of what each one of these look like. Before I get into the detail on those, I want to talk a little bit about terminology. Even people that know the correct words, people that should know the correct words, this terminology can get tricky. And I also spend a lot of time talking to people about what is the right method for their project. And people can feel very strongly about one method is better than the other and you shouldn't be doing the other. The right method for you is project specific, and quite often it comes down to what can you do for data analysis and how much money do you have? So let's say you have a mixed community. You extract all the DNA. Should you do amplicon sequencing, so targeted PCR products, or should you do metagenome sequencing and sequence all the DNA? For amplicon sequencing, meta barcoding, that is targeted sequencing of specific regions. For example, 16S for bacteria or ITS for fungi. That's going to be less expensive per sample. You can get more samples multiplexed per run, and it is easier to analyze. If you are working with undergrads or new grad students who are new to bioinformatics, this is usually the way to go. The data is a whole lot easier to analyze. For metagenomics, where you are going to shotgun sequence, you're going to break apart all the DNA in that sample. When you go to put that DNA back together, it is really complicated. You need to be good at bioinformatics. So when people tell me they want to do metagenomics of a mixed community, the very first thing I'm going to ask is, are you good at bioinformatics or do you have someone to help you? It is also more expensive per sample. Each sample has to be sequenced at a much greater depth of coverage. However, the benefit is you are not limited to whatever you targeted with the amplicon sequencing. I love this slide. I didn't make it, I added to it. But this is a conversation I have a lot with people. There is no right or wrong answer as to what is right for your project, but it usually comes down to cost and your bioinformatics skills. Whoops, I touched things. There we go. So the next 3 slides, I'm going to go through our most common workflows. All There are multiple ways you can do all three of these workflows. The diagrams that you will see are pulled from what we do in our lab. Doesn't mean this is the only way to do it. For whole genome sequencing, as I mentioned, this is the process of determining the entire DNA sequence of hopefully a single organism's genome at one time. Examples would be you might be doing this to document phylogeny or look for variations within a population. Metagenome sequencing would be sequencing the entire genome of every organism in a mixed community. Almost any whole genome or metagenome library prep protocol, the first step you're going to do is you have to fragment the DNA in some way. You have to break it apart. This is either going to be done enzymatically or by physical shearing. Then some way you have to ligate on those adapters. If you look at the bottom of this slide on the left-hand side, that image should look familiar to you when I said every sequencing fragment needs to end up looking the same. In my workflow there, it says limited cycle amplification and indexing. Somewhere in there, you're going to add all the pieces so that we can get it looking just like that, like all pieces need to look at the end. Amplicon sequencing or metabarcoding. Now, there are multiple ways you can do this. This is the sequencing of specific targeted regions of the genome by multiplexed PCR. So we're going to do a whole lot of samples by PCR. We are looking at in-depth analysis as quickly and cost effectively of a whole lot of samples. And typically requires less sample input than the other sample types. So lots of reasons you might do this is to rapidly screen many samples for what bacteria are present. The method that we use in our lab is a two-step PCR. We start with a locus-specific PCR that has adapters on them to make the second step work. So we do the first PCR, we purify it, then we do an index PCR that adds on those other pieces. Around the middle of this slide, you can see it has those same multiple pieces on it. And then we do another purification. You can put all these pieces on in one initial PCR, but it is a complicated PCR and it requires very expensive initial primers. The reason I don't do that is it's not very universally applicable for me in the core. By doing a two-step PCR process, I can apply that to whatever PCR primers you might want to use for your research. The last most common method we do is RNA-seq. I will be totally honest with you and tell you this is my least favorite method. I like DNA, which is easier to work with, but also this is the most expensive thing we do. Any method of RNA-seq, like I said, is going to first convert it to cDNA. Total RNA-seq sequences all the RNA present in a sample. You can measure gene and transcriptome abundance. You can detect known and novel features in coding and non-coding RNA. And you can targeted hybridization can remove abundant rRNA to focus on high-value proportions of the metatranscriptome. mRNA sequencing can quantify gene expression. Transcriptomes can look at all the mRNA within a mixed sample. And RNA enrichment analyzes gene expression in a focused set of genes of interest. This diagram on the left is the kit that we use. So we can do the first strand cDNA synthesis. We universally deplete the rRNA. And then we're back to that same process we talked about before, where we're adding in all the pieces that we need to get the final product looking like all the other library preps. So to give you an example of something we do in our lab that I thought might be applicable to this group, this is one of my favorite examples of something we did, partially because it was working with leatherback sea turtles, which is just fun. There was, I don't know if Deb Miller made it on or not, because this is her student who did this many years ago. Researchers at the vet school were investigating the bacterial microbial profile on the skin of leatherback sea turtles. The student was looking at neonates at emergence, neonates a few weeks of age, and then nesting females. The first time she sent me her samples, almost none of them worked. One of the benefits of our lab, we want your stuff to work. So she did her extractions herself. She sent us the extracted DNA. Then we took over and we did the PCR and the library prep. Well, after that initial PCR, I emailed her and I said, hey, this did not work well. Let's talk about what we have. I learned some about her project, and when I started talking to her, I realized certain types of samples worked better than others. So we were able to have a conversation. I was able to put her in contact with someone who was taking similar swabs so we could talk about, are you using the best swabs for your method? Are there better swabs you could use that might collect more DNA? We were able to talk about, oh, hey, some of these samples might not actually have much bacteria and they may actually fail because they don't have any bacteria. By coming to us, we want to help students. That's what we are here for. We want to troubleshoot. We also did some troubleshooting of her PCR and got more of them to work. So again, this is one of my favorite projects because there were many things going on. It was a first-time student doing extractions. I also know this particular project was done in the Florida Keys where they lose power a lot, and some of her samples sat in freezers where the power went off and she didn't know the freezers had lost power. So we had a lot of things going on, but we were able to help the student prepare amplicon sequences, and she got papers out of it, and the student is now working she's come back to me to ask for help with her own students. So I thoroughly enjoy projects like this where we can help troubleshoot and still get some really neat results out of it. All right, so let's talk about some of the additional services that we offer. I mentioned earlier, I just came from teaching. One of the things that we offer is training in lab techniques. We encourage people to come into the lab and take tours of the lab, and when appropriate, work with us in the lab to learn the techniques. But I also offer a class. It is cross-listed as Micro 679 and an undergraduate level class cross-listed through Micro and EEB. For undergrads, it must be upper level undergrads, and they have to have my permission to join. There are some prerequisites, but honestly, the most important one is that they know how to pipette. The goal of this class is just to teach students the hands-on lab skills they need so that they can do these projects on their own. The feedback I get from most students is that really valuable to learn the hands-on skills that often their PIs may not have time to teach them. We also assist with project design. I consider it my job to take the time to sit down and talk students through what maybe they might not feel comfortable talking to their PIs about. When students come to me and they say this, I have a dumb question, I try to set up my lab as an environment of we truly have no dumb questions. And if you feel like you have questions you maybe are embarrassed to share with your peers or with your PI, come to us. We want to help you. We are a safe place for those questions. We also do all of our ordering. I used to say this is a benefit, but it's all done through a software that automatically connects to your account. Now that it's through Dash, I know this is still a headache, but hopefully we will get to where this is less of a headache. We have a tiered pricing structure for UT users. We have a middle tier that kind of lumps anybody that is not commercial, and then we have a commercial tier. I wasn't sure if we'd have any ORNL users on here. ORNL is tricky. If you have a joint appointment where you have a UT account, if you are paying with your UT account, you get UT pricing, but if you have to pay with an ORNL PO, it counts as that non-UT academic rate. But I do try to work with ORNL users to figure out the best way to get through their projects. I will tell you it takes forever to get an ORNL PO to getting ORNL and UT to talk is one of the hardest things I do. We work on a first come, first served basis. It is really helpful to know when projects are coming. We have an actual queue. You're not in the queue till your samples are in my hands, but you do get in my mental queue, and it's helpful to know when your projects are coming. Some of our minor equipment is available for users to use on a minor user fee, but the major equipment, such as these sequencing instruments on the right, are only used by the core staff just to make sure that they stay in good working order. But we do encourage users, particularly students, to be present and see how they function as appropriate. Benefits of using University Core. The number one most important one is you can take your samples and put them in my hand. You remove both the scare and the cost of shipping precious samples. I knock on wood every time I say this, but we've never lost a sample pre-processing. I have had people come back 3 years later wanting their samples back. I can't guarantee that, but we do a pretty good job keeping track of samples. We also are excellent at troubleshooting. We care about every sample. Even if you do the library prep yourself, if it fails on my instrument, that makes me look bad. And I want to know why it failed. If you send it off to a big box company and your samples fail, they don't care. I care. I take pride in every sample that goes on my instrument. We also are going to tell you if we get to the final QC and we don't think it's good enough to proceed. We're going to give you the chance to say whether or not you want to. Sometimes people will say to me, I know the QC looks bad, but that's all I have. Give me the data you can. But I'm going to let you make that decision. We also often attempt samples that other larger facilities have rejected. You would be surprised how many times we can get them to work just by diluting them. So bring us your samples that are tricky, that you think might fail. We're willing to try them, and we want to. If everything works right the first time and we are just processing samples, that is boring, and that does not make for enjoyable work. Now, with that said, the flip side of that is also true. I don't want samples that require troubleshooting every single time. Sometimes we get so many difficult samples that I look at my technicians and I say, somebody please just bring us a sample that's going to work right the first time. New and upcoming offerings. If you have talked to me before, you may know that my prices for DNA extraction were very high. That was because DNA and RNA extractions take a long time. Well, I now have enough staff that we have been able to drop our prices. I think our prices for DNA extraction are pretty good. We now have a DNA extraction robot that is not being used as much as I would like it to. With that said, not every sample is meant for automation. If you come to me saying you want to automate your samples, we're going to have a discussion about whether or not they're right for automation. I do not like soil samples, soil or fecal samples being automated. But there are a lot of sample types that can be automated to save you money and me time. So let's talk about extractions. We also, I mentioned early on, if you remember, we have gotten some new smaller rooms, and one of the uses of these rooms are a shared bench space. If you are someone that does not have your own lab to work in, we offer this as a clean room area that you can come. It is a small user fee, $10 an hour, more if you need assistance from us. We are working on getting equipment for that room, but we plan to have a PCR machine, centrifuges and vortexes. As you see in the picture, it has pipettes. We will offer non-filter tips at no cost, but if you need filter tips, we will have to charge for those. We are also working on getting a qPCR machine that users can use for a small user fee as well. We are going to be offering DNA and RNA quantification services. If you ever use the ones in the former sequencing facility in Mossman, these are not going to be where you can just come and quantify your own. We will be using our instruments. So we want to make sure we maintain the integrity of the instruments, but we currently have a NanoDrop 8. We're working on getting a qubit that can do more than one sample at once. But we're going to be offering these as services that we can do for you. And we are hoping to add a tape station to SURF in the very near future. I think we're going to be able to do that in the new fiscal year. Another thing that we want to do, we want to know when you have particularly interesting results from our sequencers. I occasionally get asked for projects we can spotlight. I am not going to brag about your sequencing results unless you tell me I can, because that is not my project to speak of, unless you tell me you want me to. I am working on getting a way I can my words are not coming to me, but put papers in the hallway in front of my lab. So if you, particularly for students, if you have a publication out using sequencing data, send it to me and I will put it in front of my lab so people can see it. This includes theses and dissertations. But please remember, it is beneficial to me and keeping the core facility moving. If you can reference the lab in your methods or in your acknowledgments, as simple as sequencing was done at the University of Tennessee Genomics Core. Bringing new instruments into the core, we are always open to conversations. If there is something we don't have that you would like to see, if it makes sense for it to add to our core, if it's going to be useful to multiple users in multiple techniques, we are open to those conversations. Usually requires some kind of financial buy-in from interested users or grant proposals, but we are open to those conversations. For example, we currently do not have a third generation long read sequencer. These tend to have higher error rates than what we currently have because they sequence so quickly. Examples being PacBio and Oxford Nanopore. However, this is a giant however. PacBio just released a new instrument that is at a reasonable price range that I am very interested in getting into my core if there are enough people that are interested in using it and that would actually come use it. So if this is something you're interested in, let me know because I'm starting to put together a list of users that might be interested in it to see if it's worth me pursuing. I mentioned we don't do data analysis, but we do collaborate with 2 groups on campus that can help with that. Our data is too big coming off of these instruments for us to take care of ourselves. So OIT's bioinformatics group helps with that. And a group that can help with bioinformatics consulting is Meg Staten and Ryan Custer's group. I have a slide for each of them. The bioinformatics computing support group, here are some links to their websites. I encourage you to get on their newsletter. They offer Zoom office hours. As I mentioned, they do all of our data transfer. Our data coming off of our big instruments is stored on UT's cluster. It is securely stored and you can access it there for quite a while. And you can also do data analysis there. And then this is a very busy slide, but just so you know they exist. If you need help with data analysis, Meg Staten and Ryan Custer's group are fantastic to at least help you get started if you need help. There's their information. So this is a very busy slide. I love it though. It shows 3 different kinds: first generation, second generation, and third generation sequencing. I encourage you to come talk to me. Let us see if we can help with your projects. When you go to price out different sequencing facilities, it is hard to compare apples to apples. It is hard for me to compare apples to apples with prices. You might be surprised if you've been using a facility for a while. Maybe we are cheaper, maybe we're not. Our prices are as low as I can get them and still make the Office of Research happy. But our prices are not meant to take your money. They're meant to just recover our costs and still keep things as low as we can for our researchers. But let us help with your projects. Come talk to me, see what we can do. Let us help with your project design. We are here to help, and we are especially here to help students. We want you to understand what you are doing. Come talk to us. And with that, I will take any questions. Thank you for having me here, by the way.
Thank you for joining us. I knew nothing before, so that was very informative. Everyone should be able to unmute themselves if you have a question you want to ask Veronica, or if you want to put something in the chat. We'll give everyone a few minutes to maybe formulate their questions. Anybody?
Yes, I have some questions.
Okay, yeah, go ahead.
One is on the gold standard for sequencing. If someone is to run sensitivity and specificity of the different platforms, what would be the gold standard they could use? And then the sequencing of viruses, because you talked about bacteria and fungi mostly. Have you also sequenced viruses? Then the other is on the training in lab techniques that you're giving for the micro 479, 679, and the EB 479. Do you plan to offer these courses in the fall, in the summer? When do you offer them? And are they strictly lab techniques or full bioinformatics courses? Yeah.
Okay, that was a lot of questions. Make sure I get to them all. Interrupt me if I don't. Okay, I'll start with my class since I'm sitting here in a classroom. I offer that every semester, every spring and fall semester. If anyone is interested in taking it, I can tell you the spring semester tends to fill up and I have to there's usually a waiting list and people tend to not get in, but the fall semester tends to be easier to get in. We do amplicon sequencing. Students go, they do their own extraction, they do the full library prep, and then we load it on the instrument. Then we do a very brief bioinformatics. I am not the person to go in depth on bioinformatics, but also you can't do in-depth bioinformatics and also get through the lab work. So we just skim the surface of bioinformatics. I try to have someone come in every semester that teaches a bioinformatics class just to say, hey, if you want more in-depth, here's where you can go. Gold standard for sequencing. That is a hard question to answer. And the answer is it depends on your project, which I know is not a great answer, but it really depends on what you're needing. All of the iSeq that I have right now, that really, when I call it my baby sequencer, it's my baby sequencer. But the NextSeq and the NovaSeq are both phenomenal sequencers, and they give you great data. For long read sequencers, if someone had the money to get me a PacBio, I would take a PacBio. Honestly, the reason we don't have a PacBio, up until recently, it's been kind of niche sequencing, and the people that wanted it, I did not feel would actually bring me their samples, and it was such an expensive instrument that it was too expensive to take the risk of users not actually coming to use it. But now that PacBio has one out that is a price range I could actually attain, I would take a PacBio if someone would get me a PacBio. And I've forgotten what the third question was.
Yeah, the third question was, I don't know.
Oh, viruses. I do not have a lot of experience with viruses. I just haven't had many people come in wanting to sequence viruses. I am happy to work with you to figure out what we need to do, but I don't have a lot of experience with viruses.
Yeah, thank you. And these classes, you said during the fall and the spring, are they the same?
Yes, same class.
Because I saw one had micro 471 and then this had EEB 479.
Mm-hmm.
And then the other had 679.
It's just cross-listed.
Oh, I see.
Sometimes students have specific needs why they need it under micro or EEB, just as a way to get more students the credit they need.
I see. Thank you.
You're welcome. Anyone else?
Sorry again, I'll ask about these
That's okay.
classes because I'm interested in taking them next fall. So are they classes that are covering much only lab techniques or general bioinformatics?
So it is just one class that is listed 3 different ways. So the first 2/3 of the class is lab techniques and the last third is bioinformatics.
Right.
But
And
again, we skim the surface of bioinformatics.
how many credits are these?
3.
Oh, thank you.
It is Thursday mornings for 3 hours. So I'll be honest, by the time I get teaching, I'm really tired and you guys are lucky. I think I made it through.
So I booked around 6:30. I could cover.
Thank you.
You're welcome. I know I'm a little biased, but I think it's a good class. We put out a survey a few years into the class and asked students what they got out of it the most. And I was surprised by the answer. Most of the students said while they appreciated the NGS side of it, many students don't have access to learn how to do DNA extraction or PCR. And I was surprised by how many students said that they benefited simply from being taught how to do and how to troubleshoot DNA extraction and PCR. So I'm very proud of the class that we are getting through to students that need that. I am glad that I am able to help students with that. And the reason we do PCR in the class is because of the different techniques we could use. PCR is a good combination of cost, success rate, and good amount of hands-on time. Genomes are just too easy. That would only be 1 or 2 days of hands-on time. I've chosen PCR. We do 16S amplicon analysis in class. Anything else?
After the
And
training.
the email that you're providing here, that is Genomics Core, is it a personal email? If I wrote to you, does it go to the core group or it goes to you?
Yes, that'll reach me. It also goes to my technicians as well, but that does reach me.
All right. I don't see any more hands up or questions in the chat. This will be posted on our website, probably this afternoon or by tomorrow, in case you want to rewatch it or share it with a colleague. And with that, I will thank Veronica Brown again for joining us. It was very informative, and we really appreciate your time, especially after that marathon class this morning. So
I
thank
appreciate
you
y'all
very
having me.
much. Everyone, please join us next month on April 24th. And I will just bid everyone a good afternoon.
Thank you and have a good afternoon.
Thank you.